Good evening, everyone. Welcome to BioTalk, the show where we unravel the wonders of the biological world. I'm your host, Izai, and tonight we've got a groundbreaking topic that's taking the scientific community by storm, CRISPR-Cas9. Joining us tonight are esteemed biologists Dr. Balkis and Dr. Shahira. Welcome. Now, CRISPR-Cas9 has been making headlines. Dr. Balkis, could you give our viewers a brief overview of what CRISPR-Cas9 is and why it's causing such buzz?
Thank you, MC. I am Nuraina Balkis. So, what is CRISPR-Cas9? It is a unique technology that enables geneticists and medical researchers to edit part of genome by removing, adding, and altering section of DNA sequence. So, genome editing is a group of technologies that give scientists the ability to change an organism's DNA. Doudna and Charpentier, the Nobelists, have published their first paper announcing new enzyme for editing the DNA in June 2012. It was called CRISPR-Cas9, which is short for clustered regularly interspaced short palindromic repeats. That consists of two components, which are Cas9 protein that can cut the DNA, and another one is guide RNA, which can recognize the sequence of DNA to be edited. So I can conclude that the existing of CRISPR-Cas9, the ideal tool for gene editing technology, we can bring the bright future for treating disease by permanently correcting delirious base of mutation or disrupting disease that causing gene with great precision and efficiency. As it is simple to use, cheap, and that makes it revolutionary. So because it could treat, perhaps even cure the genetic disease, this could really change a lot of things in society.
Fascinating. And Dr. Shahira, what is the advantages and limitations of CRISPR-Cas9 on Streptococcus aureus?
Hi everyone! First of all, thanks to the moderator for having me here to talk about the CRISPR-Cas9 on Staphylococcus aureus. Okay, let's get started. According to Chen et al. 2017, one of the common human pathogens, Staphylococcus aureus, is responsible for the high hit rate associated with infection. Human infection caused by the bacteria Staphylococcus aureus can range from the minor skin infection to more serious illness like bloodstream infection or pneumonia. The significant pathogenicity and multi-drug system for this bacteria are its sources. Unfortunately, the common genetic modification tools used to investigate the molecular mechanism of Staphylococcus aureus has several limitations, including long experimental period, relatively low gene editing efficiency, and leaving a scar in the genome as stated by Liu et al. 2017. Hence, the CRISPR-Cas9 system has been extensively employed in a variety of organisms, including bacteria as a genome editing tool. The reason is that the CRISPR-Cas9 can provide fast and efficient chromosomal manipulation in Staphylococcus aureus. In addition, as mentioned by Liu et al. 2017, eliminate the TGD and ROCK8 gene and introduce the ERM-R cassette into Staphylococcus aureus, serves as an example of how the CRISPR-Cas9 can function. Eventually, the CRISPR-Cas9 genome editing tool in Staphylococcus aureus is established by this research, it allowing for quick, colorless, and marker-free genetic manipulation.
Thank you, Dr. Shahira. Now, without further ado, we're going to welcome our three biologists to explain more about the procedures on CRISPR-Cas9, and the video has been prepared by Ms. Sherry, Ms. Ayn, and Ms. Hafiza.
Thank you, Izzat, for your invitation. So, I will give the explanation on how we perform this CRISPR-Cas9 on Staphylococcus aureus. So, for the first step, we need to construct the pCAS9 plasmid. So, how we want to construct it? So, first thing first, we will amplify the RPSL promoter from the genomic DNA of S.Aureus. This can be harvested from the pCAS9 plasmid. This RPSL functions in encoding the Cas9 nuclease. Next, we will amplify the temperature-sensitive origin, REPF, which is needed for the plasmid curing after editing it. And lastly, what we get from the S.Aureus, we will obtain the chloramphenicol resistant marker that can be harvested from PCO-R1 plasmid. Another bacteria that we use is Escherichia coli. From E. coli, we will amplify the replication origin, which is the COL-E1 and the kanamycin resistant gene from the p-CRISPR plasmid. All the 5 fragments, 3 from the S.Aureus that have been mentioned before, which are the RPSL promoter, REPF, and chloramphenicol resistant marker. Another 2 from the E. coli, which are the COL-E1 and the kanamycin resistant gene, will be assembled together into a plasmid via the Golden Gate assembly. Before resulting in final pCAS9 plasmid, we will insert the G block. G block is synthesis from the CAP1A and the synthetic guide RNA, sgRNA, into the XHO1 site of the previous plasmid that I have been explained before. And at the final plasmid, we will introduce the 20 base pair spacer of AGRA at the spacer insertion site before we transform this plasmid into the S.Aureus. So this is how the pCAS9 plasmid will look like. So that's all from me, and I will pass the next procedure to Siti Shahira.
Ladies and gentlemen, I am pleased to introduce myself as Ms. Sherry, and today I will be discussing the concept of electroporation process. Electroporation is commonly used to introduce Crispr Cas9 components into S.Aureus cells. This facilitates the genome editing process by enabling the delivery of genetic material into target cells for precise modifications. A single Staphylococcus aureus colony with a volume of 1 ml was cultivated overnight and then diluted. The resultant culture was incubated at a temperature of 30 degrees Celsius in 100 ml of TSB. Once the culture's optical density at 600 nanometer reached a range of 0.3 to 0.4, the cells were cooled on ice for a duration of 10 minutes and subsequently collected by centrifugation at a rate of 5,000 revolutions per minute (RPM) for 5 minutes. Following the removal of the liquid supernatant, a careful pipetting of 20 ml of sterile ice-cooled 0.5 molar sucrose solutions was performed to suspend the cells once again. This process of centrifugations and subsequent suspensions was repeated twice. Then, portion of 50 microliter of the cells were rapidly frozen using liquid nitrogen and thereafter stored at a temperature of -80 degrees Celsius. Finally, the cells were resuspended in a 1 ml solution of 0.5 molar sucrose. And the utilization of recently completed cells may also lead to the formation of additional colonies. Following the aforementioned steps, the cell was subsequently mixed with 1.2 microgram of plasmid DNA and transferred into a 1 mm electroporation vat at room temperature. Subsequently, the cells were deposited into a TSB agar plate that was supplemented with chloramphenicol, subsequent to undergoing electroporation at a voltage of 21 kV per centimeter, a resistance of 100 ohms, and a capacitor of 25 microfarads. Following the electroporation process, the cells were placed in an incubator set at a temperature of 30 degrees Celsius, submerged in 1 ml of TSB for an approximate duration of 1.5 hours. Consequently, the agar plate was incubated overnight at the temperature of 30 degrees Celsius.
I will continue to the next step, which is hemolysis assay. After a clone of the Staphylococcus aureus in TSB at 30 degrees Celsius overnight, the culture of strains was loaded onto a sheet flat plate. Then, the plate was observed after a 24 hours incubation period at 30 degrees Celsius. Lastly, real-time PCR is performed. In this step, 5 microliter of culture of Staphylococcus aureus strain was incubated at 30 degrees Celsius. When the optical density reached 1.0, the cells were harvested by centrifugation and lysed by fast prep at room temperature for 40 seconds. Next is RNA isolated, which is usually by using RNA ISO class. By using prime script RT reagent kit, the RNA was reverse transcripted to cDNA. For quantification of cDNA, light cycler 96 system with SYBR premix X-Tech2 kit was used. With that, it will complete the steps.
Absolutely. It's a complex landscape. CRISPR-Cas9 holds immense promise for treating genetic diseases, enhancing agricultural practices, and even contributing to conservation efforts. However, ethical concerns about gene editing in humans and ecological implications in all altering organisms should be carefully navigated. Well, viewers, buckle up as we embark on this enlightening journey to the world of CRISPR-Cas9. Stay tuned for in-depth exploration and some surprising insights. And you're watching BioTalk.